Here we are looking at an image taken from a microscope slide of blood. And this blood is a little different. This contains sickle cells. Now, sickle cell is a recessive genetic disorder. A lot of times it's referred to as sickle cell anemia because the patient typically winds up anemic due to the lack of the ability to carry oxygen efficiently. And essentially, what happens in sickle cell anemia, there is a mutation in the gene that codes for hemoglobin. And if you think back a little bit into AMP1 when you were learning about cells and protein synthesis, so you know that all the information for making proteins comes from DNA. And what happens is that information in DNA is copied into messenger RNA. That process is known as transcription. And then that messenger RNA leaves the nucleus and heads out to the ribosome. The ribosome interprets the instructions and links the amino acids together in just the right way to form a functional protein. And that part's called translation. Now, in the case of sickle cell anemia and all other genetic disorders, there is a mistake in the genetic code. That instruction, the instructional bases in DNA, if you remember adenine, thymine, cytosine, guanine, those are those four main bases in DNA. Well, in a genetic disorder, those letters, those bases, adenine, thymine, cytosine, guanine, they're out of order. There is a mistake somewhere in the code for the instructions of making a particular protein. And in the case of sickle cell, that mistake happens in the genetic code for hemoglobin. So when the cell begins to synthesize hemoglobin, which is a protein that carries oxygen, that hemoglobin is not formed properly. It's abnormally shaped. And since red blood cells carry tons and tons of hemoglobin, that abnormal hemoglobin makes the red blood cell become abnormally shaped. And it takes on this crescent shape, or what we commonly would call a sickle shape. And that's how the name sickle cell came to be. It's by the shape of that red blood cell. And since the hemoglobin is abnormal, it can't carry sufficient amounts of oxygen. And that's why a patient with sickle cell is anemic. So sickle cell anemia is the full name, usually what we refer to this genetic disease, or just sickle cell. So what we're looking at here is a blood cell sample where some of the red blood cells are normal and some of the red blood cells are sickled due to that sickle cell anemia trait. What you need to be able to do is distinguish between the normal cells and the sickled cells. So if we look closely here, we can see there's actually many normal cells. A normal red blood cell is going to be nice and round. It has indentations on each side, little dips. They're described as being biconcave discs. I typically describe them as Lifesaver cream savers. If you've ever eaten a Lifesaver cream saver, they have these dips, right, on each side. That's really what a red blood cell looks like on the microscopic level. And there are many normal ones here on this microscope slide. I'm just going to draw some circles around some normal ones to draw your eye to them. So we've got one here. This is a good one. Here's a good one. Lots of collections of good ones all around here in this area. So all of these that I'm circling, we're just going to call these normal. You can read my handwriting there. Normal, okay? Now, if we keep looking at the slide, we'll see there's some that are quite odd. They don't look like those nice round red circles. Look at this one right here. You see that one? Look at this guy down here. Right there. And look on further down. If you start looking, you can see some of these red blood cells look more like crescents or maybe little bananas, or they also make me think of like a fingernail, the edge of a fingernail that's there. These cells are your sickled cells. So what's happening is that hemoglobin is being synthesized abnormally because of that mutation or that change in the DNA. And since the hemoglobin is abnormal, the red blood cells are ending up abnormal as well. So we're going to identify these. I'm just going to draw some lines since I have them spaced out pretty far here, these are sickled. Okay, so we have our sickled cells that look like those crescents or sickled shapes, and then the normal ones are the nice round plump red blood cells. Okay, now on test, what I'll do is I'll ask you to distinguish which cells are normal, which cells are sickled. So be able to pick out those shapes when you're identifying a normal versus a sickled cell.